respiration, which is a fundamental characteristic of every living organism. It is a catabolic breakdown of a suitable substrate so as to generate a usable form of energy. Now, the substrate at times could be a simple carbohydrate like glucose or fructose, or it could be a fat molecule. In such cases, the process is called as floating respiration. Whereas sometimes even a protein molecule can be broken down to generate energy when it's called as protoplasmic respiration. When I say usable form of energy, it's the ATP molecule which I'm talking about. Now, ATP molecule is an adenine bonded to a pentose sugar that becomes adenosine, to which 3 phosphate groups are bonded. So that becomes adenosine triphosphate or ATP. Now, in this ATP molecule, the bond between second and the third phosphate is known to be an energy rich bond or a high energy bond. That actually means when the third phosphate from ATP is detached, this bond energy between second and third phosphate is liberated, and it is this energy which living organisms can make use of for their routine activities. So, usable form of energy, when I say, I'm talking about an ATP molecule. After giving out the third phosphate, what remains behind is an ADP, adenosine diphosphate. Now, the respiratory mechanism can take place in presence of oxygen when it's called as aerobic respiration. Sometimes it can take place in absence of oxygen when it's called as anaerobic respiration. Now, efficiency of a catabolic breakdown would be decided on the basis of number of ATP molecules it can generate. And from that point of view, aerobic respiration can generate 38 molecules of ATP, that is equivalent to 673 kilocalories, whereas anaerobic respiration can generate just 2 molecules of ATP. So, aerobic respiration is quite an efficient process of respiratory breakdown as compared to anaerobic process. The aerobic process involves 3 main phases. The first one, it's called as glycolysis or EMP pathway, Embden-Merov-Parnas pathway, that takes place in the cytoplasm. Second phase is Krebs cycle, or it's also called as TCA cycle, tricarboxylic acid cycle, or citric acid cycle. This takes place in the mitochondrial matrix. While the third phase, actually it's not the third phase, it's a phase which runs almost parallel to Krebs cycle or even some part of glycolysis. It's called as electron transfer system, that's terminal oxidation. It takes place in the F1 particles present in the mitochondria. Now, to understand the position of matrix and F1 particles, let us have a look at the internal structure, or let's say ultrastructure of a mitochondrial component. Let us see what a mitochondrion looks under microscope. As could be seen, it's a double membrane bound cell organelle. Inner membrane is thrown into a number of folds which are called as cristae, and suspended in the matrix is a separate DNA and ribosomes. That is why it is called as a semi-autonomous component of a cell, almost independent of the cell. In addition to that, as could be seen here, on cristae are located certain particles which are called as F1 particles. Now the ultrastructure shows that there's a base, there's a stalk which is called as F0 portion, and there's a knob-like thing that is called as F1 particle or oxysome. That is the site of electron transfer system.